If you want to predict who is going to develop colon, breast, or pancreatic cancer in the next 10 years, don't look at their family tree. Don't look at their cholesterol panel. Don't even look at their colonoscopy report. Look at one specific bacterium hiding inside their gut wall right now. It has a name. It has a face under a microscope. And there is a growing pile of research from Harvard, Yale, and the Dana-Farber Cancer Institute showing that this single microbe is acting like a personal bodyguard for cancer cells, shielding them from your immune system, fueling their growth, and even disabling chemotherapy when patients try to fight back. The shocking part? You are feeding it 3 or 4 times a day with a food sitting in almost every American kitchen right now. And once I show you what it is, you'll never look at your pantry the same way again. For decades, doctors told patients the same thing. Cancer is mostly bad luck and bad genes. Eat clean, exercise, hope for the best. That story is falling apart because in 2011, a team of researchers cracking open colon tumors found something nobody expected. Living bacteria woven directly into the cancer tissue. Not contamination, not an infection, a bacterium that had set up camp inside the tumor and was actively helping it grow. That bacterium has now been linked to colorectal cancer, esophageal cancer, pancreatic cancer, and aggressive breast cancer. And the question stopped being, does our microbiome influence cancer? It became, how do we starve the microbe that's driving it? That's exactly what I'm going to show you in this video. I am a physician, and for the last 10 years, my clinical and research focus has been on the gut microbiome, specifically how the bacteria living inside us either protect us from chronic disease or quietly drive it. And I'll tell you straight, what I'm about to share isn't fringe. It's not a wellness theory. It's coming out of peer-reviewed journals like Cell, Science, Nature, and Gastroenterology. And almost nobody is talking to patients about it in plain English. That changes today. In the next 15 minutes, I'm going to walk you through 5 things. The exact name of the gut bacterium that may be feeding your cancer, and how to know if you're at high risk. The 2 specific mechanisms it uses to protect tumors and disable your immune system. I'll keep them short, clear, and easy to remember. The one food you almost certainly eat every day that scientists at Yale proved acts like rocket fuel for this bacteria, and why low-fat labels often hide it. A 6-food anti-tumor protocol with exact daily amounts based on human studies designed to starve the bad microbe and rebuild your protective bacteria. And finally, 3 pro tips and 3 common mistakes I see patients make when they try this on their own. Stay with me till the end, because the last food on the protocol list is the one almost nobody mentions, and the human data on it is genuinely impressive. Before I dive in, one quick thing. If somebody you love has been diagnosed with cancer, or if cancer runs in your family, drop the word family in the comments below. I read these. It tells me what topics to dig into next, and it also signals the algorithm to push this video to more people in the same situation. That tiny gesture genuinely helps somebody else find this information at a moment they need it. Okay, let's get into it. The bacterium I want you to remember is called Fusobacterium nucleatum. Most people have never heard of it. Your doctor probably hasn't talked about it. But in the cancer research world, it has become one of the most studied microbes of the last decade. Here's the strange part. Fusobacterium nucleatum normally lives in your mouth. It's part of the dental plaque that forms on your teeth between cleanings. In a healthy person with a healthy gut, that's where it stays. The acid in your stomach and the friendly bacteria in your colon keep it out of places it doesn't belong. But in people with a damaged gut lining, weakened immune defenses, or a diet that constantly inflames the intestinal wall, this microbe slips through. It travels down. It finds spots in the colon where the barrier is thin, and it embeds itself directly into the gut tissue. Here's the analogy I want you to hold on to. Imagine your gut lining as a brick wall protecting a city. The bricks are your intestinal cells. The mortar between them is a protein called E-cadherin. As long as the mortar is intact, nothing dangerous gets through. But Fusobacterium has a tool, a surface protein scientists call fada, that dissolves the mortar. Once a section crumbles, the bacterium walks right into the deeper tissue, and once it's inside, your immune system has a much harder time getting at it. Why does this matter? Because in 2012, two independent research teams, one at the Broad Institute at Harvard, one at the BC Cancer Agency in Canada, sequenced colon tumors from hundreds of patients. What they found stunned the field. Tumor tissue had up to 400 times more Fusobacterium than the healthy tissue right next to it. The bacterium wasn't randomly there. It was selectively enriching itself inside the cancer. And follow-up work has now found this same pattern in esophageal cancer, pancreatic ductal adenocarcinoma, a particularly aggressive subtype of breast cancer, and metastatic colorectal cancer that has spread to the liver. The same Fusobacterium DNA shows up in the original tumor and in the secondary tumors. Translation: the bacteria are traveling with the cancer cells. They're hitchhiking. They're part of the metastasis. So why is your body letting this happen? That brings me to the two mechanisms I promised you. I'm going to keep them short, because once you understand these two, the rest of the video makes complete sense. The biofilm cloak. The first trick Fusobacterium pulls is what I call the biofilm cloak. A biofilm is a slimy, protective layer the bacteria can build around themselves. Think of it like a fortress wall made of sticky sugars and proteins. Dental plaque is a biofilm. The slimy layer inside an unwashed water bottle is a biofilm. When Fusobacterium nucleatum sets up shop inside a tumor, it doesn't just sit there. It builds a biofilm over the tumor. Once that protective layer is in place, two things happen. First, your natural killer cells, the immune cells whose entire job is to find and destroy abnormal cells, physically can't reach the cancer. They bump up against the biofilm and get blocked. Researchers at Memorial Sloan Kettering filmed this happening under microscopy. The immune cells are right there, present, but disarmed. Second, the bacterium releases a molecule called FAD-A2 that locks onto immune cells and switches them off. It's like flipping a kill switch. The immune cell stays alive but loses the ability to attack. So you have a tumor that is shielded by a fortress and surrounded by deactivated security guards. The second mechanism is about fuel, and this is where the food side of the conversation begins. Most cancer cells run on sugar. That's well known. But what scientists at Yale discovered in 2019, published in the journal Science, is that one specific type of sugar acts as a targeted accelerator for tumors infiltrated by Fusobacterium and similar opportunistic microbes. That sugar is high-fructose corn syrup. In their experiment, mice with early intestinal tumors were given the equivalent of one daily soda's worth of high-fructose corn syrup. They didn't get fat. They didn't gain weight. The amount was carefully controlled to rule that out. But the tumors in the corn syrup group grew dramatically larger and more aggressive than tumors in the control group. The mechanism the Yale team described works like a highway. Fructose hits the gut. The liver doesn't fully clear it because the dose overwhelms the system. And the excess fructose circulates and gets handed off directly to tumor cells. Tumor cells then convert that fructose into the building blocks of new cancer cell membranes, basically using it to multiply. Now, combine the two mechanisms. You have a bacterium building a fortress around the tumor, deactivating your immune cells, and a daily delivery of cheap rocket fuel, keeping the cancer well fed. That is the engine I want to dismantle. So let's start by cutting off the fuel. Let me give you the studies because I don't want this to feel like opinion. This is data. Dana-Farber Cancer Institute, 2017. Researchers analyzed tumor samples from 1,069 colorectal cancer patients. Patients whose tumors contained high levels of Fusobacterium had significantly worse 5-year survival than patients whose tumors had little or none. The presence of the bacterium independently predicted outcome, even after controlling for tumor stage, age, and treatment. Yale University, 2019, published in Science. Mice with early intestinal tumors were given a daily dose of high-fructose corn syrup equivalent to a single 12-oz soda for a human. Tumor size in the HFCS group was substantially larger than controls within 9 weeks. Importantly, weight gain was matched between the groups, ruling out obesity as the driver. Fructose itself was the accelerator. This is no longer theoretical. This is the food fighting the bacteria. Now, let me show you exactly how to do it. Here are the 6 foods I want you starting this week, with exact daily amounts pulled from human trials, and a sentence on why each one matters. Resistant starch from cooked and cooled potatoes or rice. 1 cup per day served cold or reheated gently. This is the workhorse. When you cook a white potato, cool it in the fridge for at least 12 hours, and then either eat it cold or reheat it under medium heat. The starch molecules realign into a form your small intestine can't digest. They travel intact to your colon, where Akkermansia, mucinifera, and other beneficial species feast on them. Akkermansia is the bacterium that thickens your mucus layer, the same layer that normally keeps Fusobacterium from getting close to your tissue. Cold, leftover rice in a stir-fry, potato salad without the mayo bomb, or simply a baked potato cooled overnight will do it. Frozen, then thawed potatoes work just as well. Ground flaxseed, 2 TBSP per day, freshly ground if possible. Flax is the highest known dietary source of compounds called lignans. Your gut bacteria convert lignans into enterolactone, a molecule that has been shown in human studies to reduce systemic inflammation and modulate estrogen-related cancer pathways. A meta-analysis from the Mayo Clinic in 2021 linked higher enterolactone levels to a lower risk of breast and colorectal cancer. Stir 2 TBSP into oatmeal, yogurt, or a smoothie. Whole flax seeds pass through largely undigested. You have to grind them. Live fermented vegetables, half a cup per day. Kimchi, sauerkraut, or plain kefir. Has to be the refrigerated kind. The shelf-stable jars at room temperature have been pasteurized and contain almost no living bacteria. You're looking for raw, unpasteurized, or contains live cultures on the label. A 2021 randomized trial at Stanford showed that adults eating 6 servings of fermented foods per day for 10 weeks had a measurable drop in 19 different inflammatory markers and a noticeable increase in microbiome diversity. Half a cup daily is a sustainable, realistic version of that protocol. Shiitake or Maytake mushrooms, 100 green, about 1 cup chopped, at least 4 times per week. These mushrooms are dense with compounds called beta-glucans, which act on a part of your immune system called the dendritic cells. These cells essentially wake up your natural killer cells and make them better at finding tumor cells. A Japanese trial published in 2020 gave colorectal patients a daily mushroom extract dose for 12 weeks and saw a significant uptick in natural killer cell activity. Sauté in olive oil with garlic. Add to soups, omelets, or rice bowls. Dried mushrooms rehydrated in warm water work just as well as fresh. Jerusalem artichokes, or chicory root coffee. a small daily serving providing 5 Dru of inulin. Inulin is a prebiotic fiber that selectively feeds Bifidobacteria, one of the most protective groups of gut bacteria. Bifidobacteria produce short-chain fatty acids, particularly butyrate, which are the preferred fuel of your colon cells and keep your gut barrier strong. A small handful of roasted Jerusalem artichokes or a single cup of chicory root coffee in the morning will get you there. Start small and ramp up over a week. Too much inulin too fast can cause bloating. Wild blueberries, half a cup per day. Frozen is fine and sometimes better. I'm putting blueberries last because they are quietly one of the most powerful foods in this entire protocol, and most people underestimate them. The pigments that make wild blueberries deep blue are called anthocyanins. A study at Tufts University showed daily consumption of 1–2 cup of wild blueberries for 8 weeks shifted the gut microbiome toward the protective profile we're trying to build. Wild blueberries, the small ones, usually frozen, have roughly twice the anthocyanin density of cultivated. Toss them on yogurt, blend into a smoothie, or eat them straight from the freezer. That's the protocol. 6 foods, real amounts backed by human data. Now, let me give you a sample day so you can see how easy this is to actually do. Here's what a normal Wednesday looks like on this protocol. Breakfast: plain Greek kefir with half a cup of frozen wild blueberries and 2 TBSP of freshly ground flax. That's 3 of the 6 foods before 8 a.m. Mid-morning, a cup of hot chicory root coffee instead of your second cup of regular coffee. Inulin done. A grain bowl built on yesterday's leftover rice, straight from the fridge, topped with a 1/4 cup of kimchi and sautéed shiitake mushrooms. Resistant starch, fermented food, and beta-glucans all in one bowl. Dinner: whatever protein you normally eat with a side of roasted vegetables, and most importantly, no HFCS-loaded sauces or dressings on top. You hit all 6 foods in a single day without doing anything exotic, without buying supplements, and without spending more than about $15 extra on the week's groceries. These are the things I tell patients in the office that you won't hear in most articles online. Reheat your starches gently, not in the microwave on high. The resistant starch molecule is fragile. Blasting cold rice or potatoes in the microwave on full power converts a chunk of it back into regular digestible starch. You lose the prebiotic effect. Either eat it cold, or reheat in a pan on medium heat, or microwave on 50% power for a longer time. Small detail, real impact. Add a fat to your blueberries and your kimchi. Anthocyanins from blueberries and certain compounds from fermented vegetables are far better absorbed in the presence of fat. A drizzle of olive oil on the kimchi side dish or full-fat Greek yogurt under the berries instead of fat-free can roughly double the absorption of the active compounds. Fat-free is sabotaging you here. Floss your teeth, seriously. This sounds absurd in a gut bacteria video, but I'm dead serious. Remember, Fusobacterium nucleatum lives in dental plaque before it ever reaches your colon. The cleaner your mouth, the smaller the reservoir of this bacterium that's available to seed your gut every time you swallow. People with periodontal disease have roughly twice the colorectal cancer risk of people with healthy gums. That's not a coincidence. Floss. Use a tongue scraper. Get a cleaning every 6 months. This is cancer prevention as much as it is dental hygiene. Let me also flag the 3 mistakes I see patients make over and over. Going too hard, too fast. If you've been eating a low-fiber diet for years and you suddenly add resistant starch, fermented foods, and inulin all on day 1, your gut bacteria are going to throw a party, and you're going to be uncomfortable for about a week. Bloating, gas, irregular bathroom trips. Add one new food every 3 to 4 days. By the end of a month, you're at the full protocol with zero discomfort. Mistake 2, replacing the food with a supplement. I know it's tempting. There are pills marketed as resistant starch capsules and fiber blends and polyphenol concentrates. Almost none of them produce the effects measured in the food-based trials. Whole foods bring in dozens of cofactors that the isolated supplement leaves out. Eat the food. Trying this for 2 weeks and giving up because nothing changed. The microbiome doesn't shift in 2 weeks. The Stanford fermented food trial took 10 weeks to see the full effect. Real microbial change is on the order of 3 to 6 months. The good news is, once it shifts, the new state is fairly stable as long as you keep eating in this pattern. This is not a 30-day diet. It's a way of feeding your second brain, the one in your gut. I want to leave you with this. Cancer is not a verdict written in your DNA at birth. Family history matters, yes, but it accounts for only a fraction of total cancer risk. The bigger story, the one that's emerging in journal after journal right now, is that what you eat over years and decades shapes the ecosystem inside you. And that ecosystem decides whether the microscopic abnormalities that form in everyone's body get cleared out quietly or fed and protected until they become a diagnosis. The patients who beat the odds aren't doing anything magical. They're not taking exotic supplements. They're not on cleanses. They are quietly, consistently feeding the right bacteria and starving the wrong ones day after day, meal after meal. The defenses are already inside you. The protective bacteria are already there, waiting to be fed. You just have to stop poisoning them and start feeding them. You can begin tonight. Cold rice from the fridge, frozen blueberries, a spoonful of kimchi. That's it. That's the start. Let me leave you with a quick recap so this sticks. The bacterium to remember is Fusobacterium nucleatum. It uses 2 main mechanisms, a biofilm cloak that hides tumors from your immune system, and a sugar highway that turns added fructose into fuel for cancer cells. The single most important thing to stop today is added high-fructose corn syrup. And watch out for the emulsifiers, polysorbate 80 and carboxymethylcellulose. The 6 foods to start: cooked and cooled potatoes or rice, 1 cup. Ground flaxseed, 2 tablespoons. Live fermented vegetables, ½ a cup. Shiitake or maitake mushrooms, 1 cup 4 times a week. Jerusalem artichoke or chicory root for inulin, 5 gd daily. And wild blueberries, ½ a cup. And don't forget, floss your teeth, reheat starches gently, eat a little fat with your antioxidants, and give the protocol at least 3 months. You have far more control over this than you've been told. Your microbiome can be reshaped. The defenses inside you are real. They are measurable, and they respond to food faster than to almost any pharmaceutical we have. Here's what I'm going to ask you to do. If anything in this video shifted the way you think about cancer, send it to one person tonight. Just one. A parent, a sibling, a friend who's been struggling. 1 in 2 men and 1 in 3 women will face a cancer diagnosis in their lifetime. And the information you just learned could change which side of that statistic somebody you love ends up on. And if you want to go deeper, my next video will be about the markers that quietly predict cancer risk 5 to 10 years before any tumor shows up. It's the natural follow-up to what we just covered. Take care of your gut. Take care of the people you love. And I'll see you in the next investigation. If this story changed the way you look at the microscopic world within your gut, please subscribe and like the video. We are uncovering the biological truths that have been hidden for too long. Let's cultivate a healthier future together. The next breakthrough is coming soon.